Hi, my name is Benjamin Isaacoff, and I'm a graduate student at the University of Michigan studying applied physics. Today I'm going to tell you about how we are peering into the nanoscale world with the help of tiny optical antennas. Optical microscopy, which just means using microscopes to look at stuff with light, is a ubiquitous technique throughout all of science. You're likely to find a microscope in just about every lab that you look in, and moreover, this is true ranging across nearly all of science, engineering, and medicine. And in fact, for many of those labs, microscopy is an absolutely integral tool to everything that they do. Microscopes are useful because they allow us to see small things. And no, I don't mean small like the bottom of the line in the eye chart. I mean really small, like things we can't see with our eyes alone. For example, these plant cells are about 30 microns wide, or about 1/3 the width of a human hair. But microscopes can easily see much smaller things than that. These bacteria cells are about 1 micron wide, or 1/100th the width of a human hair. And amazingly, if you look closely at this image, you can see some internal features of the cells, meaning that we can get information from this image on a length scale smaller than 1 micron. Maybe even the smallest features on this image that we could identify would be about 500 nanometers. As is clearly evident here, microscopes are a truly incredible tool. Part of the reason that optical microscopy is so ubiquitous is that in addition to being so incredibly powerful, they are quite easy to use and understand. School children regularly use them. They're relatively cheap and easy to acquire and even move around. And importantly, in comparison to more high-tech methods of microscopy, optical microscopes are non-invasive and non-destructive to most every sample, greatly expanding what can be studied with the technique. But even though optical microscopy allows us to see tiny things like these bacteria, it can sometimes be very unclear what those tiny things are, and more importantly, what they're doing. Imagine looking at a cell with a lot of complicated stuff going on inside. For example, these black lines could represent proteins moving around inside a bacteria cell. Trying to pull out information on a specific protein, for instance the protein that moved along this path, would be like trying to follow a needle as it moves around in a chaotic haystack. It would be rather difficult. Thankfully though, researchers a long time ago came up with a brilliant and simple solution to this problem. They realized that the needle would be a lot easier to follow if it was emitting light, or more specifically, if it was fluorescing. In fluorescence microscopy, scientists attach a fluorescent molecule to the object they want to study and use that fluorescence to watch it. In this way, scientists can get out incredibly specific information from microscopy. But how does fluorescence work? Well, let's start out with our fluorescent molecule. And fluorescence is a two-step process. First, we need to add energy to the molecule. We need to excite it. This is typically done by shining a laser or some other kind of light on that molecule. This is our excitation event. Now that the molecule is excited, it can relax in energy by emitting light. This is fluorescence. So with optical microscopy, we can see really small things, and with fluorescence, we can study it with really high specificity. But what's the limit? Can we see arbitrarily small features? It turns out that the answer is no, and the reason for that is a fundamental part of optical physics. As this animation, reminiscent of Pink Floyd's Dark Side of the Moon album cover, is demonstrating, light is a wave, and that's the problem. Waves behave in ways that can be somewhat counterintuitive. To illustrate why that is a problem for imaging really small things, let's imagine an infinitesimal point source of light. A physical process of waves called diffraction will always spread that infinitesimal point source into a blurry spot whose size is dependent on the wavelength of that light. So for visible light, that diffraction-limited spot will be about 300 nanometers across. This limitation on imaging small things can be a big problem. For instance, if you want to study a really small bacteria cell, you're not going to be able to get much detail out with 300 nanometer resolution. But what some, now very famous, scientists realized was that this diffraction process was very well understood. They could really predict how it works for a single point source of light. And if they could somehow come up with a way to image just single molecules one at a time, then they could effectively sidestep the diffraction limit. And this realization led to the development of single molecule fluorescence microscopy. By looking at just one single fluorescent molecule at a time, understanding how diffraction blurs that image, and some clever mathematics, those scientists realized that they could locate where that single fluorescent molecule was with very high precision, even surpassing the limitation imposed by diffraction. Then to make an entire image, they just repeat that procedure one molecule at a time until the entire image is complete, analogous to pointillist paintings. Using this technique, scientists were able to increase the resolution of optical microscopy by an order of magnitude. Whereas before the smallest details that could be made out were about 300 nanometers, they can now easily get details at 30 nanometers. And using this technique, scientists can now learn so much more than they could have previously. For example, this is an image of a gut bacteria which has some proteins of interest fluorescently labeled. On the left is the conventional fluorescence microscopy image. You can't make out many details due to diffraction, but on the right, using single molecule fluorescence microscopy reveals unseen details in how those proteins were arranged in the cell. Because this technique and related ones have had such a tremendous scientific impact in so many fields, the researchers who developed it were awarded the 2014 Nobel Prize in Chemistry. By bringing optical microscopy down to the nanoscale, this technique has enabled scientists in fields ranging from biology and medicine to material science and chemistry to make groundbreaking discoveries and push science and technology into unprecedented territory. Now, there is no doubt that this is an amazing technique, but in theory, the technique should be able to provide resolution of objects at a nearly 1 nanometer. However, in practice, resolution is rarely better than 30 nanometers. To illustrate why, let's take a look at an image of a single molecule. First, let's consider a bright single molecule. On the left, I'm showing an image, and on the right is the intensity map of that same molecule. When the molecule is very bright like this one, it is straightforward to locate, and you can do so with very high precision, leading to excellent resolution. But if instead we have a molecule that is dim, it is much more difficult to locate, and even if you can, you won't do so with very high precision, and consequently have poor resolution. And in the limiting case where the molecule is very dim, it is almost impossible to locate, and you won't be able to image anything at all. So in order to have the best resolution, we want to have bright molecules. However, the reality is that in many experiments, we are always stuck with these dimmer molecules. In my research, we are addressing this problem. We are developing the science and technology to be able to take these dim molecules and to make them brighter. Our main strategy to do this is to utilize optical antennas. Optical antennas are analogous to the antenna in your car, but instead of responding to large wavelength radio waves, optical antennas respond to shorter wavelength visible light, the light that we see with our eyes. Although this concept isn't difficult to imagine, it turns out that the best optical antennas are nanoscale structures made of metal. And it wasn't until the last decade or so that nanotechnology has matured enough for us to really start studying and utilizing them. In particular, one of the optical antennas that I have worked the most with is a nanoparticle shaped like a rod and made of gold, shown here with an electron microscopy image. From the scale bar, you can see just how incredibly tiny these optical antennas are. I think a useful definition of optical antenna is that it is a device that can convert free propagating optical radiation to localized energy, and vice versa. Now let's go through this definition in detail to make sure that we really understand what an optical antenna is and how it can enhance single molecule fluorescence. Free propagating optical radiation just means light. Importantly, the kind of light that we are used to in everyday lives. It is freely moving through space. It is propagating. Now in practice, you don't just have one light wave, you have a lot. So what happens when you put an optical antenna in the path of all this light? Well, as the definition tells us, it converts that free propagating optical radiation into localized energy. It does that by absorbing a huge amount of the incident light. Not all of it, but quite a lot of it. And through some remarkable physics called a surface plasmon, the antenna converts that light into localized energy. Now what about the vice versa direction? What happens if we have a localized source of optical energy? This basically just means a light source that is spatially very small. This is exactly what our fluorescent molecules are, but it could be any small light source. What happens if we place an optical antenna near this localized source of energy? Through that same remarkable physics, the presence of the antenna causes the source to actually emit a lot more light. It may also reshape or redirect that light, but importantly, the antenna is converting the localized energy in the source to free propagating light. With this understanding of how an optical antenna works, it is straightforward to see how it could be used to enhance single molecule fluorescence. By converting free propagating optical radiation to localized energy, we can enhance the excitation of a fluorescent molecule, then allowing that molecule to emit more light and appear brighter. By converting localized energy in the fluorescent molecule into extra free propagating optical radiation, we can directly enhance the emission of a fluorescent molecule, making it appear brighter. And this is really great. Just by using one device, the optical antenna, we can enhance both steps of fluorescence, both the excitation and the emission of a single fluorescent molecule. And by doing so, we can make even very dim molecules appear brighter, and thus we can locate where the molecule is with much higher precision, resulting in unprecedented levels of resolution. This is still an extremely active area of research, because it turns out to do this most effectively is really complicated. There is a plethora of obvious questions that need to be answered. For example, it's really important to think about the colors involved. Does your antenna work in the red, but your molecule is blue? How far apart should the molecule and the antenna be? Surely it shouldn't be really far away, but it turns out that if it physically touches the antenna, it doesn't fluoresce at all. Also, fluorescent molecules actually have a well-defined orientation, and it can be really important to consider how that orientation matters, especially with respect to the antenna. And of course, there are a million things to consider with regard to the antenna itself. When should you use a gold rod versus, say, a copper ball? Or is a silver star a better choice? Or what about something that is half one material and half another, and so on? We've been working very hard to try and figure all these things out, to determine a complete physical theory of this system. We are doing a lot of experiments. These are actually lasers that I use every day in lab, and also working from a theoretical perspective as well. And I'm confident that we will eventually get there, that we will develop a full theory, and with that insight, be able to design the best possible optical antennas to enhance single-molecule fluorescence microscopy, a technique which already improved the resolution of optical microscopy by an order of magnitude. And with the aid of these tiny antennas for light, we may be able to improve the resolution even further and open a window for all scientists to peer into the nanoscale world.